All right. Well, I think I'm going to get started. I want to make sure I leave enough time for our speaker today because we have an excellent talk for everyone today. Thanks everyone for joining us for the May webinar of the Boston Epigenetic Society. I have just a brief introduction before we get started. As you may know, the mission of the Boston Epigenetic Society is to promote epigenetics and epitranscriptomic research and to foster connections between academic and industrial epigenetic scientists. And we have an excellent leadership team that helps us put together these presentations. I have the people indicated over on the left, and I really want to thank our sponsors, especially Active Motif, who's generously helping us share the video today, the webinar. And also Cantata Bio helps us extensively with that. And then 10x Genomics and Biomodal are also fantastic supporters of the Epigenetic Society. If anyone is interested in helping out and being a part of the Epigenetic Society and would like to help pull these activities together, please reach out to us. Our email is bostonesociety@gmail.com. And also, one thing I'm very excited about is today's speaker actually has attended all of the webinars and she wanted to share her work. So if you have work you would like to share with us, please do let us know. That's the whole point of the society, is to try and bring people together, foster collaborations, foster conversations to promote this type of research. And again, the way to do that would be to reach out at bostonesociety@gmail.com. Other ways of getting involved and connecting. Right now, our LinkedIn page is our main point of contact. So tune into that for upcoming events. And if you'd like to present or you would like to learn any more about the Epigenetic Society, we're in the process of pulling together a website and we hope to unveil that shortly. Our upcoming events, we're going to be at the Festival of Genomics at the BCEC here in Boston, June 12th and 13th. Come on down and say hi. Our next networking event is we're going to try to do something in the Seaport area on June 12th. So stay tuned. Those details are still coming together. And we have an exciting webinar planned for the summer. Details are yet to be determined, but stay tuned for that. So I don't want to take any more time. So I'm going to introduce today's speaker. The title is ProA and ProB Repeat Sequences Shape 3D Genome Organization and Eukaryotes. And as I said, this is an active listener and participant of the Epigenetic Society. Genevieve Fourel is joining us from France. So I thank her very much. And just a note to the audience, if you have questions for the speaker, please write them below in the Q&A window. And if you have questions of not related to the talk, but more logistics, you can put those in the chat window. So thank you very much. Oh, and now I do have to introduce Genevieve. Today we're thrilled to welcome Dr. Genevieve Fourel as today's webinar presenter. Genevieve's fascination with biology began early, and at 18, a summer job in the lab solidified her passion for research. Not singing, as she quickly discovered. Her academic journey took her to the prestigious École Normale Supérieure. Forgive me if I butchered the pronunciation, where a fellowship allowed her to join Jerry Crabtree's lab at Stanford University. There, she made her first major discovery, finding the first tissue-specific transcription factor, which set her on a lifelong quest to understand gene regulation from A to Z, or A to Z++ even. Back in France, she spent a decade at the Pasteur Institute, diving into prokaryotic transcription factors and liver cancer research. Her successful PhD on liver cancer led to a research position at INSERM, where she explored gene activation mechanisms and enhancers. In 1996, Genevieve joined Eric Gilson's lab in Lyon, where she enjoyed a 6-year holiday studying yeast and genome organization. Her work during this period laid the foundation for understanding eukaryotic genome organization and long-range chromatin interactions. In 2002, she became the research director at INSERM and founded the Cancer Epigenomics team at ENS Lyon, aiming to decode genome organization and its role in cancer. Despite funding challenges, Genevieve's determination never wavered. After a decade-long hiatus from the lab, she returned in 2017 to continue her groundbreaking work on repeat sequences and genome organization, thanks to her global dream team of bioinformaticians. Please join me in welcoming Dr. Genevieve Fourel, who will share her inspiring journey and pioneering research with us today.
Thank you, Brian, for this long introduction, and I will jump into my presentation to keep time on track. And so today I will present my work. Sorry, I will try to deal with these windows. So I will present our paper, which is available on bioRxiv, and I will also take time to present some earlier works that were carried out actually at the time I was working with Eric Gilson. And because what I'm presenting today is actually a lifelong project which involved many, many people. Sorry. Whose name you can see on this slide. Sorry, I have to adjust to the presentation. And nothing of what I will present today would have been possible without my dream team of bioinformaticians, as Brian mentioned, who are dispersed throughout the world, and especially Constantin Bonnet, an amazing student. I am very much indebted also to Camille, which represent the contribution of the scientific community, which is huge in our case because we use essentially data that were produced by the community. So when you look at the eukaryote cell under the microscope, it is known for very long now that you can see heterochromatin, a dark part of chromatin and the nucleus, and euchromatin, which is light. And I was very surprised to learn recently that it was already known at the time of Emil Yates in the early 30s that heterochromatin contains repressed genes, whereas euchromatin contains many more genes. And most of the genes which are expressed are in euchromatin. Therefore, heterochromatin is an environment which is unfavorable to gene expression, whereas euchromatin is favorable. We, as a community, are now pretty good at describing the arrangement of chromosomes in a nucleus. And this is thanks in particular to amazing techniques which are based on superior resolution microscopy. All these papers have been published in the past 5 years, many actually coming from labs in the Boston area, but also papers from Europe. But before that, we had already learned a lot thanks to the C techniques, which are based on cross-linking, freezing interaction between two segments of DNA using a cross-linker, which is usually formaldehyde. And therefore, what we monitor with these techniques is the probability of interaction of two DNA segments in the nucleus. This was carried out initially with plasmids, then with single locus, and eventually there was a genome-wide version that I'm sure you all know, and which is known as IC, this famous paper by Job Decker's group. What IC told us is that each chromosome can be viewed as a mosaic, a succession of domains belonging to the A and B compartments, which actually more or less correspond with euchromatin and heterochromatin. And therefore, the arrangement of chromosome in the nucleus should be understood as a folding of this kind of organization and producing in the end this type of image. That's what heterochromatin and euchromatin is in the end. However, one thing you should not forget at this stage is that these IC techniques, at least the data that we'll be using, were carried out using cell populations, and therefore this image eventually, this mathematical image of genome organization correspond to an average image of the individual organization in each of the cells. This is what physicists call the mean field representation of genome organization. So this is given by the IC eigenvector profile, which I will now explain a little bit more about. So this is the IC eigenvector shown in brown, in blue, for the human chromosome 10. And what I'm showing here is an enlargement of a 25 megabase region, and I will show it several times in my talk. The reason why I chose this region is that you have a big 5 megabase E domain. So this corresponds to euchromatin. And the fact that it's euchromatin is indicated by the fact that the value for the IC eigenvector are positive. And therefore, this is a B domain corresponding to heterochromatin. So you have these two big domains, and then on the right part, you have all these small domains, and you cannot miss that the value of the IC eigenvector, which corresponds to the contact probability, is lower for the small domains compared to these big domains. So what this means is that, for example, in these small A domains, a segment found here will have a lower probability to interact with another A segment than in this big domain, where the segment will tend to interact all the time with another A fragment. So this slide shows a comparison of the profile of the IC eigenvector for 8 cell lines, which are ENCODE reference cell lines. We'll be using UVAC in the rest of this talk because it's actually the only primary cell. It's not a cell line, and I want to insist on this. These are differentiated cell, endothelial cell of the umbilical cord. Whereas all these are really cell lines, some of them derived from cancer. And I think you will agree with me that what is most striking at first sight, the general impression, is that the general profile is conserved. Of course, it's not true in detail because it is known that the IC eigenvector is, as I said, a mathematical image of genome organization, and genome organization and gene expression are proper to each cell type. So of course there are differences, but the general impression is that of conservation. And if you try to analyze why you have this impression, it's because of these two big domains, which are actually more or less conserved in all cells. So this domain is always A, and this domain is more or less always B, and that's how we will call them. Whereas these small domains often tend to shift between A and B, and that's the way we will call them from now on, A or B domains. And you cannot miss that these were also small domains in UVAC. So this also contributes to this general impression. And what the fact that you have this general conservation tells us is that there is something that is common between the different cells and that play an important contribution to the IC eigenvector, in other words, to genome organization. And what is common between these cells? It is their DNA. So there must be some DNA determinants of genome organization. In addition, what this tells us, the fact that you have these small domains which are smaller everywhere and have low value of the IC eigenvector, is that these cis DNA determinants not only determine whether a domain will have a strong tendency to be either A or B, but it also determines whether domains will display some plasticity. This is just a quick slide to say technically that we slice the genome. We use HG19 genomes for an early version. We slice the genome in 100 kb bin, and then we put these bins in 24 chromatin subclass based on their chromatin color, the gene density, and whether the bin displays or not active promoter marks. And these correspond to bin counts in high density bins, regular density, and in bins where you don't have any genes. And it's obvious that in the HG19 version, at least, the most important part is this panel, the regular gene density. This is another representation of the same data. And I think it is obvious and striking, actually, that each of these chromatin class represent approximately 25% of the genome. So we set out to identify the cis determinants of AB partitioning. We didn't start from scratch. Actually, very long ago, we used YEST, the YEST Saccharomyces cerevisiae, to study AB partitioning. And we showed that the forces that drive AB partitioning in yeast actually obey this scheme. And this scheme is what system biologists call a toggle switch scheme. What does this mean? That we should envisage AB partitioning as a system with two individual chromatin subsystems, A corresponding to the type of chromatin you find in the A compartment, and same thing for B. There are forces that are responsible for mutual opposition between A and B, which means that once you have an A chromatin type chromatin in one place, there will be force that will repel B from installing. And these arrows mean that there are self-reinforcement forces, some mechanism that makes each of the stats somewhat stable, sufficiently stable so that we can indeed sometimes see A or B. Otherwise, we'd be all the time switching between A and B. And this is all described in this early synthesis article of mine. This might still look a bit abstract to you, so I will take time to illustrate what it means. In all organisms, the B compartment depends on heterochromatin. So what is heterochromatin? It's a system of components of which there is, in this system, you have a principal component, which is the heterochromatin structural factor protein, which is called SIRC in yeast. And in whole heterochromatin, the structural components bind to nucleosomes and compact the chromatin fiber. However, there is this mark which is found actually in the whole eukaryote, H4 K16 acetylation. In all eukaryotes, in the A compartment, And this mark opposes binding of nucleosome by CRC. Therefore, there is a component in the SIR complex, an enzyme which is called SIR2, that removes the green mark such that CRC can bind to nucleosomes. You see here that you have a euchromatin enzyme opposing an heterochromatin enzyme and vice versa. That's mutual opposition. And a euchromatin histone mark, which is removed in heterochromatin, again, opposition. In addition, because the SIR complex will compact the chromatin fiber, it will prevent access to the enzyme that are responsible for the presence of the green mark in the A compartment. Therefore, this is again a layer of mutual opposition. Actually, this is just a small piece of all the mechanisms that are responsible for mutual opposition. It looks more like this, and there are all these active marks which are well known, but I just ask you to remember the principle of mutual opposition with enzymes in euchromatin and heterochromatin and histone marks, which are more or less a similar roles, but which are in opposition. CRC binding to heterochromatin in yeast is actually prominently observed in proximity to heterochromatin nucleation center, which correspond to transcription factor binding to the DNA, which is not explicitly represented here, and which recruit the heterochromatin structural factor. And one important piece of information here, of course, is that the SIR complex interacts between each other and through multivalent interactions. Therefore, the heterochromatin nucleation center recruit SIR complex and recruit also the SIR2 enzyme, which will modify nucleosomes such that CRC can bind, and more SIR2 enzyme will be recruited, which again modify nucleosome, and more CRC can bind. And so what you're seeing here is what we call heterochromatin spreading. And because SIR complex interacts through multivalent interaction, in the end, you will have a kind of a cloud of SIR factor, which are anchored at the chromatin fiber through the SIR factor, which are spread. And in addition, because the SIR factor can bind many partners, you will end up with a volume of the nucleus with a different composition from the rest of the nucleus. Not only you will have this SIR complex playing the role of a scaffolder, condensate-like structure, but you will have all these partners also, which will be enriched. This represents the self-reinforcement force, and maybe you've already realized that this read-write mechanism, whereby the CRC factor bind, recruit the enzyme, and then allow for the binding of more CRC, which recruit more enzyme, is typically a self-reinforcement mechanisms. But actually, there are many more of these mechanisms that altogether make this cloud quasi-stable, and such you can, that if you follow the system over time, you will see a certain stability, which doesn't mean that it's totally stable. It's still highly dynamic, but it is quasi-stable. So actually, I hope you better understand what I mean now with this. Oh, sorry, I forgot to say that. Of course, this drawing is in 1D, but in reality, it is in 3D. And this artistic drawing is just to show you the vision that we have of what this silent making type cassette in Cerevisia look like. And this is just a 3 kb region, so a very small region. And you don't have to think that everything is all crowded, all packed. It doesn't mean All this doesn't mean that there are necessarily a lot of this factor. It's just that the probability of presence is increased and the factor have a tendency to stay together. So I hope now you have a clear idea of what this scheme means, this toggle switch scheme. But actually, in this article, we went a bit further and we showed that the heterochromatin nucleation center actually promote the formation of the B compartment in yeast. They are what we will call from now on ProB elements. And same thing on the green side, on A, these gene regulatory elements play the role of ProA elements. They keep the region free of heterochromatin. And what are these cis elements? In yeast, they are essentially the promoter of genes and some elements that at the time we called insulators and that protect the region from the spreading of heterochromatin. And all this idea of toggle switch has been largely confirmed since. But let's try to forget all these details and try to imagine a strategy to identify the ProA and ProB element in human, just based on this understanding of the toggle switch scheme. Also based on the fact that we know that we have this IC eigenvector that provide us with a mathematical image of genome organization. The always A region display a very high value of the IC eigenvector, so they have a strong A character, and therefore in this region, you would expect to have a lot, really a lot of these ProA elements that promote the E compartment. And when I say this, actually what I'm saying is that we expect to have a correlation between the density of the ProA elements and the value of the IC eigenvector. And something on the B side, expect that here we would expect a counter correlation with the IC eigenvector. In addition, these ProB elements on the A side are expected to destabilize the A compartment because of this mutual opposition. Therefore, they will tend to be selected against in the A compartment. So along with evolution, we'll tend to lose them and we will end up with a strong enrichment between here and here, which means that in the B compartment, you expect to have an enrichment of these ProB elements. So we end up with these two criteria regarding the density of this ProA and ProB element, correlation with IC eigenvector and enrichment in A or B. In addition, if we indeed found such type of sequences, just out of the density of the ProA and ProB, we should be able to predict back the IC eigenvector. To make a long story short, we focused on repeat sequences, simply because genes represent only 2% of the genome approximately, whereas repeat sequences represent, they cover 45% of the genome. This is for the HG19 version, but actually in the full genome version, the T2T, it's 56%. So they're definitely the main component of the genome. Regarding transposable elements, you find Lyon, in particular Lyon 1, endogenous retroviruses, signs, for instance, ALUS. I should mention that the Earths in the genome mostly correspond to solo LTR because of recombination between the homologous LTRs. And in the genome, the line one are the most abundant, then come the signs, then come the errors. As a matter of comparison, recently, with the advance of really high throughput techniques, DHS, which are DNase1 hypersensitive site and correspond to the enhancers, were found to represent a class of elements with a similar number of as repeat sequences, and I will come back to this later in my talk. Actually, there are not only transposable elements, but also sequences which are not, repeat sequences which are not transposable elements. But I will have little time to mention them in my talk. Therefore, we took this 1,395 repeat sequence subfamily, which are found in the red-based database. And for each of them, we determined the density in each of the bins of the HG19 version of the genome. And then we determine for each of the subfamily the correlation with the value of the IC eigenvector in the bin and the enrichment in A or B compartment. The correlation will be taken for significant beyond this 0.01 value. And we decided that empirically on the threshold for the enrichment in A or in B. And therefore, these two squares are the place in our figure where we will find dots for subfamilies which are the criteria we are looking for, which are both correlating here with B and enriched in B, and this name CoreB enriched B and CoreA enriched A here. And here is the result. So that's a lot of dots, 1,395 corresponding each to one subfamily, lots of colors, but still you can see trends. Here, a lot of peaks, a lot of pink at the tip of this horn. These are line ones. And I'm asking you to remember that these are actually young line ones, these subfamilies which are called L1 PAs. On this side, in the CoreA enriched A square, you have all these green dots which correspond to ALU subfamilies, and also some of the blue dots corresponding to SVA, which I forgot to mention in the previous slide. It's a very low abundancy family, actually a class which is proper to human. Herbs are shown in red, orange, and yellow, and they are mostly in between, but also in this CoreB enriched B square, which is better seen on this figure, where you have the copy count of TE here for the full genome with the age indicated by age class. And here are the CoreB enriched BT, and you see that it's about approximately half L1 and half Rs. And the CoreA enriched A are mostly ALU, and the L1 are actually all L1. And if you look at the age, it is striking that you have more green here than here, which means that in these two sets of repeat sequences, there is an enrichment for very young and young sequences, which is a strong argument to say that what we're looking at here is really specific, and you need an intact sequence because transposable elements decay with age, the sequence drift, If you, for example, want to keep a transcription factor binding site, you will be more likely to have it in young sequences than in old ones. If we look at the distribution of these repeat sequences along the genome, the core-enriched A in green and COB-enriched B in red, well, I should say it's as expected because you have a lot of green here overlapping with the big A compartment, a lot of red here, and the profile more or less follows the profile of the IC eigenvector, which is indeed expected. I take time to comment a bit about the chromatin feature that we'll be using in the following. A growth sac corresponds to the transcription activity. And indeed, you see that transcription is essentially observed in the A compartment, which is exactly what we expect. DHS, so DNase 1 hypersensitive site, the enhancers, also, this is a density in 100 kb/bin, and again, you see a close overlapping, a close correlation with a profile of the A domain. And this is just for an illustration concerning the B compartment. This is the Easton marks, which is required for the binding of HP1 alpha, which is a structural factor responsible for heterochromatin in human. And indeed, it is also characteristic of the B compartment, as you can see here. So one prediction was that we would be able to predict genome organization just out of our repeat sequence set. Therefore, we decided to do linear models. And I will start by commenting the models we did just with chromatin feature. So this is the IC eigenvector, always on the same region for illustration. And this is the profile produced by the computer as a prediction of the IC eigenvector, just using GrowSAC as an input. And you see that the prediction, as indicated by the red square value, is very high, is more than 75%, 100% being the theoretical maximum value obtained by combining all features. DHS is a good predictor, although not as good as GrowSAC. And strikingly, if we look at the model obtained with taking only the density of repeat sequences and the input, we also combine gene density, therefore considering gene as a special type of repeat sequences in a way. Strikingly, the prediction is almost as good as the one obtained with DNase 1 hypersensitivity density. And I want to stress the point that this is really striking because DHS is a chromatin feature. It will be, the profile of DHS will be different in different cell types, but these density in repeat sequences are DNA features. They are identical in all cell types, and you end up with almost as good a prediction, which is really amazing. And if you combine both, you end up with a very good prediction. So you may tell me, but again, this is all expected given the way we define those set of sequences. And I will say, yes, you're right. Indeed, we have sequences that correlate, that are enriched, and so we expect to be able to do that. Therefore, what is remarkable is the fact that the A and B compartments are sufficiently heterogeneous with regard to their composition in repeat sequences such that we are able to define this set of repeat sequences and be able to do that. Another representation of the distribution in the genome is this box plot representation where we represent the density in individual chromatin class. A chromatin class with the highest A character is this one, which is always A, high gene density and activity. And one with the most B character is this one, always B, no gene and no activity. And I think you will agree with me, it is striking that we have a kind of a linear progression with more and more ProB from year to year and less ProA. So we are also verifying this hypothetical drawing that we did at the beginning. Some may still be convinced that this is whole coincidence and this is all expected. Well, it is my opinion to say that when I say this, what comes to my mind as the most parsimonious explanation is that the relative proportion between ProA and ProB sequences in any bin will be responsible for the tendency of this bin, trend the proneness of this bin to be part of either A or B compartment. So please keep this in mind. Early in our analysis, actually in the first time we did this figure, we early noticed something that we thought might be interesting, is that the DHS density in these small domains was strikingly as high as is in always A domain. Whereas intuitively, we would have expected to have a lot of this DHS here because this region is high gene density, and we also did a Go analysis, and we know that in this kind of domains, you have housekeeping genes which are strongly active. So we were expecting a lot of these DHS here. I will better show you this by showing an enlargement of this region. So this is the A domain, and actually it's pretty difficult still to see the DHS at this enlargement. It's more easy to see them with this alternative histone mark, which is H3K4ME1, which is also found at DHS or active enhancer. But again, I will enlarge to focus on this 400 kb region. And here you can see this peak corresponding to DHS. And strikingly, you have a peak here, 1 year, 1 year, 1 year. These are small ones, but you have them more or less occurring regularly along the domain. So these guys overlapping with H3K27 acetylation are classical active enhancers, but these small peaks are what is called primed enhancer. And this identity of DHS that I'm illustrating here, we actually did a rigorous analysis, and we were able to show indeed that in small A domains, you have a strikingly high density of DHS. And in addition, what this broad distribution suggests is that DHS are a kind of a force that cooperatively repel B compartment to open domains. Another piece of knowledge that comes to support this idea are observations that were done during development. So this represents an ideal gene which is expressed at some stage during development. It depends on the promoter activated by enhancers. But earlier in development, it is not expressed. Actually, it's embedded in the big compartment. The only thing you see is the H3K9M3 marks and not really anything else. And at some point, the cell will decide to express the gene. For example, it's a beta globin genes, and the cell will differentiate and turn into a red blood cell. and the cell will decide to express the gene, and the genes will therefore end up in the A compartment. But the interesting point here is that there is an intermediate stage where the gene is not yet expressed, but the locus is already part of the A compartment. It has increased in volume. And what is remarkable from the chromatin point of view is that DHS have appeared. They are primed enhancer in the sense that they don't have the H3K27 acetylation mark that you find at active enhancers. And therefore, what this strongly suggests is that primed enhancer, simple DHS, are sufficient to cooperatively open domain. I want to mention also that during this process, the enhancers that were initially methylated at CPG lose methylation. In the past 10 years, little by little, with the advance of DNA-seq and more high-throughput techniques, there were papers that were published and that reported that in one specific tissue at specific stages during development, a class of enhancers were actually corresponding exactly to a class of repeat sequences. Well, this is actually how transposable elements were discovered, because a transposable element inserted nearby the gene that was responsible for kernel colors, and that's how Barbara McIntosh initially identified transposable elements. And I think that the real demonstration that all DHS enhancers are really of repeat sequence origin is in this paper by Bing Ren group that was published at the end of 2023. Therefore, we should consider that the core A enrich A, the core B enrich B, and the DHS enhancers are one single system of repeat sequences. And our vision of what happens is that along a domain, you will have constitutive ProB repeat sequences, so the core B enrich B, and convertible repeat sequences, which are enhancer. And at some point, a number of these convertible ProB will convert to a ProA state, an active enhancer state. And when a sufficient number of these will be green along a domain, it will open the domain and generate an A domain. This is also represented here in the toggle switch scheme. The A compartment takes over and repel the B compartment. Another intriguing observation we did early just with this browser view was this one. So this is a UVEC IC eigenvector, and this is actually a lymphoblastoid cell line, which is among the ENCODE reference cell line. And we notice that in this region, almost all the changes compared with UVEC were switches from B compartment to A compartment, which are indicated here with all these green squares. By comparison, this cell line, IRM90, which is also used by many labs, display changes mostly from A to B compartments, which are indicated here in red. You may not know what is a lymphoblastoid cell line. So these are actually B lymphocytes that are obtained from a donor, and you infect them with the virus Epstein-Barr in vitro in a dish. And this results in the B lymphocyte becoming a kind of an immortal cell line and indefinitely multiplying. I tried to find a picture of an LCL and I couldn't find one, but I will show you a photograph of a B cell stimulated by an antigen because my virologist friend always told me that this was what an LCL looked like, a B cell which has been stimulated by an antigen. And I borrowed these images from this amazing paper by Raphael Cazel's lab. So this is a B lymphocyte, the same kind of cell I showed to you at the beginning. It has a small nucleus, the chromatin is highly compacted, and few hours after stimulation or infection by Epstein-Barr virus, the nucleus has increased in volume in a very impressive manner. It's massive, and in addition, the chromatin is highly decompacted. And this makes sense when you see this profile that you have a massive shift from B to A compartment. But this was characterized in details by a number of labs in the past years. And what all these labs found is that the Epstein-Barr virus encodes for its own viral transcription factors, and the viral transcription factor will combine with B-cell transcription factor to activate enhancers, but these enhancers are actually repeat sequences. So again, we're left with this picture with viral transcription factor and B-cell transcription factor switching ProB sequences to a ProA state. Interestingly, this was a wonderful system to characterize a switch from a ProB state to a ProA state, and they could show that it's much more subtle. For example, there were cases where you could already see a bit of H3K4M1, like these small enhancers, small prime enhancers that I showed to you, and after the viral infection, these are switched to full enhancer chromatin state, active enhancer chromatin state. So this would correspond to this. But you would also have case when the sequence was clearly in a ProB state and then it would switch in a bit less ProB state. Therefore, we should revise our picture to present the switch of ProB to ProA like this, which indicate actually that every repeat sequence in the genome should be considered as a composite of ProA and ProB capacity. What are the molecular mechanisms? I told you, if you remember, that the core B and rich B that were really at the tip of the core were the youngest of the L1. What do they have in particular? Well, the very young T will be the one which are the most repressed by the heterochromatin machinery, because you don't want to have them expressed, you don't want to have them recombining because they are very similar sequence. So it's very important that they are very well repressed. So what this tells us is that the ProB sequence very likely exerts a ProB function by playing the role of heterochromatin nucleation center. And therefore, repressing these young Ts is both a necessity for the cell to keep its genome intact and also an opportunity to organize its genome. On the E side, don't have time really to argue, but we have evidence that these active ProA elements, I mean, the convertible repeat sequences in the ProA state, actually act to repel the heterochromatin spreading. And I will come back later on the constitutive ProA. You certainly notice that this is not very different from what it found in years, almost 20 years ago. An important aspect here, though, is that those sequences individually, they can do nothing. If we see their activity in the genome, it's because there are many of them, they exert their function only by cooperating with sisters and brothers and cousins, such that in the end, it is the relative proportion of the red guys and the green guys that tells us whether you're in the A or the B compartment. I want to stress here that the pink shading represents heterochromatin spreading, and this cooperativity between ProB elements, which is fundamental to understand this cohesion in the B compartment, is likely underpinned by the condensate-like properties of heterochromatin. And ProA elements, I mean convertible sequence in their ProA state, likely function by enriching euchromatin component and creating a kind of cloud, in particular enriched in euchromatin enzyme, that will repel the B compartment through this mutual opposition principle that I exposed earlier. And finally, I will say a word about how we think that ALU exert their ProA activity. It's not very well known, although it's very well published, that ALU sequences are the place in the genome where the nucleosome are the most positioned. And we think that this positioning actually opposes, is not totally preventing, but it opposes somewhat heterochromatin spreading, such that when you have a lot of them, it will be almost impossible that heterochromatin establishes, which will result in always a domain. In conclusion, I hope you're now convinced that repeat sequences, much more than genes, drive genome organization in eukaryotes. And I illustrated with this infection with Epstein-Barr virus that equipped with this understanding, we can now explain basically all type of reorganization that can be observed in different contexts. I didn't have time to show you what we can explain with cancer, but basically, and I hope I will have time to discuss that later, I think we are able to explain how a cancer cell is endowed with its most iconic feature, I think, which is cell-fate plasticity. The driving force of AB partitioning obeys these toggle switch rules, which is true between compartments, is also true between the ProA and ProB element, and there is a very important component, which is cooperation, and eventually each repeat sequences should be considered a composite of ProA and ProB capacity, with the constitutive ProA and ProB generating a scaffold of organization which is common to all cell types, whereas the convertible sequences drive domain opening. Thank you. I should stop sharing. I cannot hear you.
I hope you can hear me now.
We hear you now.
I know we're at time, but I was hoping that we could maybe get in a couple of quick questions. This one from John Pugh. In your model making, could you find a role for small molecules which can diffuse rapidly and into any chromatin compartment, and which act on chromatin proteins in an allosteric manner, switching their role? Such molecules could be products of general metabolism with some alteration, for example, cyclic AMP or PPGPP. So their levels could be controlled via that metabolism, or possibly via the synthesis of molecules which combined the small molecules, thus competing with chromatin. Any thoughts?
Well, as I said, you have these two chromatin system, which is considered a system, and we know their components. So anything which will act individually on this component will act on each of the system, and in the end on the system as a whole. So, for example, if you think of acetyl coenzyme A, which is produced by the mitochondria, this will push histone acetylation toward acetylation and not toward deacetylation. And this will promote the E compartment and in the end promote unfolding, for instance. And but some molecules, some small molecules can also directly act on the components themselves. For example, it is known that a very small molecule, which is a product of SIR2, actually promotes the binding of SIR-C to the chromatin. So it is true that small components can themselves also have a role.
Another question, this one's my question. You had talked about Epstein-Barr virus as possibly changing the states from ProA to ProB or vice versa. EBV has been implicated with MS patients and the onset of multiple sclerosis. Have you given any thought to looking at MS patients to see if maybe this is a mechanism for MS onset?
Well, I think it is now clear the mechanism through which Epstein-Barr virus is involved in multiple sclerosis is because the EBNA1 factor, which is actually a chromatin factor, has a common antigen with myelin, and therefore this elicits an autoimmune response. So it doesn't really have to do with chromatin organization. Sorry.
Another question here from Diego Borges. How conserved do you believe this form of genome organization is conserved? Do you think it's conserved all the way down to unicellular life forms?
I think it's conserved in eukaryotes. And even, I mean, the repeat sequences would be very different between species. It's even one characteristic, but the function, the fact that the ProB will be heterochromatin nucleation factor and will be the young ones will be conserved. The allele won't be called allele in other species, but we know that for some reason having to do with this nuclear positioning that I mentioned, and which is common to all repeat sequences that derive from pol-3 RNA, this will be conserved, and these guys will play the role of ProB, ProA sequences. Actually, we'll know this very soon because of all these genomes which are characterized and sequenced these days.
And that seems similar to this question from Armel Corpat. RepSeq drive genome organization in A and B compartments, but does this mean that a gene in the B compartment will always be silent, even if it's in a different cell? Can you have a solo active gene in the B compartment?
Okay. Actually, just walking along the genome, very infrequently, you can find a gene which is expressed really in the B compartment. But still, I say in the B compartment, but sometimes the A compartment is not so far. And in this case, what you see are some very strong enhancers and many of these DNase 1 hypersensitive sites. So it is an additional support to this idea that enhancer repel B compartment, even to create a small bubble of A sufficient. I mean, we draw this like this, but in 3D, you have to imagine it can be on the border of the chromosome territory and touching some other A compartment regions. And the more we refine the IC analysis, the more we see that these I mean, this resolution we have when we deal with 100 kb bins is really coarse grain. And when you refine the analysis, you can see that actually these regions that we say have an expressed gene in B, when you refine the analysis, you realize that they actually interact with other A regions, so they are part of the A compartment. And we are biased, in a way of speaking, by the resolution of the analysis.
Thank you. Well, we're over time, but I do want to thank you again for a great talk. Very, very interested to see how this progresses, particularly as it applies to cancer. And I'm seeing some other questions pop up that people are very curious to see how this plays out as it's applied to cancer. So thank you again for your presentation. And thank you to everyone for joining us today.
Thank you so much. And this is for everybody. Don't hesitate to write to me and when we can discuss. Thank you.